I don't know, from some reason this disconnected. Sorry for interrupting you.
No worries. Should I start again or?
Okay, now it's. I think it's just as a make like.
Yeah, just a summary. We have defined regions based on a gene list. We went to Screen. This is a database with genome-wide tracks. We then went to Nomad. It's a variant database. We took the standing variation within this defined regions. And now we are at this stage with a list of variants and, of course, the associated regions. We subsampled the more rare and singleton variants and then went into the experiments nicely done by the Ahituf lab, as you can see in the next slide. But first, the MPRA is a high throughput method. You can test from a sequence of interest what the effect on the transcription initiation is within a cell type of interest. And this was the neuron I just mentioned, just to the others, where we tested it in. And if you have a reference allele and an alternative allele, you can now also quantify a variant effect within this assay. And on the right side, you can now see 3 replicate correlations of this experiment, which show that it's worked quite well. Sorry to jump back again. Before we will ask what of these variants are actually significant and how many, we will take a look in the associated reference or elements and their activity. You will hopefully learn why we go this way after the presentation. So now we ask the question, how many of our reference sequences are actually significant? And you can see here on the top right, the activity distribution from our tested CREs versus the scramble control. And in the table below, you see all the tested CREs, and around 5% were showing significant activity compared to the scrambles. They were activating and repressing effects measured. And interestingly, from screen annotations, we know that there are some known active elements, and their annotation is within our significant CRE group enriched. Yeah, the enrichment is shown here, and now we will dive into the variant effects. And here you can see of all the 37,000 variants, which we had measurements for, that we found 3% with significant effects. And we can see here the proportion of significant variants per allele frequency group, and it changed over the more rare and singleton ones, which is a little bit higher there. And I told you, you might remember that we subsampled these. And now we will look into how we subsampled these because the tested size of these variants were too high. And we used a method called Informer. It's a sequence-based method model, which can predict a lot of different experimental tracks. We were focusing only on the DNase tracks, and then we were, for each of these variant groups, we wanted to have 5,000 to test from each of these variant groups, and then we took 70% of these highly, according to the prediction, highly effect variant, 15% low effect, and 15% random. And within a group of the predicted to be high effect variants, we had a slight enrichment of only 1.7 of an odds ratio. We then heard of alpha genome. You might heard of it as well. And we try to look how much of a difference it makes if we only take, for example, the ATAC tracks. Yeah, it's a busy plot, but I hope you can make sense of it. And then also, if we take all of the features, and we see a slight improvement, especially looking for only the random sampled variants there. So it's a step in the right direction, definitely. And now, which is another thing which we figured out with looking into the proportion of significant variants is the actual reference activity matters. And you can see here again a familiar plot of the significant variants of all the references, and here filtered for only showing significant activity. And we increased the proportion of significant variants to 9%. And with that, I would like to summarize the main take-home messages. We tested, or we had readouts for more than 24,000 regions and 37,000 variants in these NGN2-induced neurons. And Former's variant prioritization showed a 1.7 enrichment compared to the significant group. And the detection of variant effects is highly dependent on the reference we look into in this kind of methods. Yeah, the most important part is the next steps. And yeah, with over 1,000 variants, we have a lot to do in terms of characterization. And I want to take the time to acknowledge all the nice people without whom this would not be possible. I'm just presenting work which was mainly done in experimental times, and therefore thanks for the Ahituv, Shenduri, and of course the Kirche Lab. Yeah, I'm happy to take questions.
Thank you very much, Kilian. If there is questions, please, from the audience.
That was great. So just when you say it depends on the reference allele, I just want to make sure I understand which direction you're going. It's not quite clear to me. If the reference allele, is it easier to detect when the reference allele is not well expressed and the mutation causes it to go up, or when the reference allele is highly expressed and your mutation causes it to go down?
I mean, there are two answers. What I showed here is only the references with a significant effect. So we need a certain effect to detect some variances. And what you also said was the distinction between upregulating and downregulating. The power increases to detect a repressing variant. When you have a higher activity, the power increases, of course, on the other side as well. Yeah. But I mean, you have this null problem that you cannot go below 0. Yeah.
Other questions?
Yes. I was wondering, so I guess your readout from the neurons was gene expression. I was just wondering if you looked at any other phenotypic readouts.
Yes, sorry, I had to make it really short, but the readout is actually, you have a barcode, you put in a sequence of interest in front of a reporter construct, you put into with lentivirus, and you have a barcode of DNA and RNA, so to say, and you only sequence these barcodes, and then you build a ratio. Like RNA over DNA is then this activity measurement, so to say. And you can then also use the reference allele and the alternative allele to then investigate the variant effects. So it's not a phenotypic readout or something. Thanks.
Yeah, I have a question. I have a question regarding the distance that you considered compared to the transcription start site, ECCB. I was wondering if there is any limitations on how far you can go, specifically because some of these regulatory sites can be megabase pairs away. And
this is not like you can test every sequences. Some groups test some scrambled sequences and like artificial design sequences. This was just our approach to look into candidate regulatory elements around these, and already the variant size within these defined regions was exploding for the more rare and singleton variants. So it depends, of course, on your setup, on your experimental, money, I guess.
Okay, thank you very much again.